Diagnosis of infectious diseases. Laboratory diagnosis of infectious diseases we can classify as microscopic examination of the clinical specimen, direct microscopic investigation, and leads to isolation using culture and its identification based on biochemical reactions, where we usually do for urine culture, bacterial blood culture, likewise. In serological identification to antigen detection and antibody immunological assays. And further, we use nucleic acid based, molecular based biological techniques. And there are other like viral isolation also available, and electron microscopy, direct visualization also a part. So first and second, one and two are often conventional methods. There are few limitations of conventional methods. Lengthy, time consuming, tedious. It takes maybe 2 or 3 days for the culture to grow. Culturing of certain like viruses, fungi, and parasites may not be possible. Some are not easy to. Viruses often not easy. Fungi may be, but parasite also same. Some bacteria also. There is also associated risk because we are dealing with live organisms. Impossible in all laboratories. Require sophisticated labs. For growth of mycobacteria, we require specific laboratory facilities. Culture may be negative due to prior contamination of prior antimicrobial therapy. That's the other one. Also, with the contamination, we can't exclude real pathogen. That's the other option, other failure. So, about serology first. Here we are not dealing about culture methods, direct tissue visualization. So, serological identification or detection of antigen or antibodies by means of immunological assays. There are important advantages. They provide early diagnosis, important for uncultivable organisms in the lab, like mycoplasma, chlamydia, useful for differential diagnosis of certain disease, useful to measure the antibody level. So there are conventional serological methods: precipitation, agglutination, hemagglutination, complement fixation, fluorescent antibody testing. Prior to that, we'll discuss some technical terms: sensitivity. We term two ways: analytical sensitivity and clinical sensitivity. Analytical is ability of the test to detect very small amount of a substance, so bacteria, fungi, or virus. Clinical sensitivity is ability of test to give positive results if patient has the disease, no false negative results. Analytical specificity means ability of test to detect substance without interference from cross-reacting substance. Clinically, ability of test to give negative results if patient does not have disease, no false positive results. Affinity. Affinity refers to the strength of binding between single antigenic determinant and an individual antibody combining site. So this is antigenic determinant and this antibody combining site. Affinity is the equilibrium constant that describes the antigen-antibody reaction. Antibody affinity is the strength of the reaction between a single antigenic determinant. So avidity is different. It's a measure of the overall strength of the binding strength. It's a strength. Okay, that's avidity is strength. In early stage of antibody formation, early formed IgM, for example, for we'll say IgG for varicella zoster or cytomegalo, their avidity is so binding force is less, but with the maturation, with the repeated production, their avidity strength of binding is high. That's how it goes. Here's some comparison other way. Multiple binding sites, avidity is high. So about we are in serological test, that is detection of antigen and antibody. So that's binding. This antigen antibody binding can be detected in various ways. Precipitation principle: soluble antigen antibody, visible precipitate. Lattice formation is there. Once antibody and antigen, they form mass of large lattice like. There are immunodiffusions, immunoelectrophoresis. Likewise, there are different tests are there. You can see multiple binding. It's about the agglutination. Antibody antigen binding leads to agglutination. Reaction of antibodies with particulate to insoluble antigen in the presence of an electrolyte at suitable pH and temperature leads to agglutination. You can see here, this is agglutination. Mesh is formed, agglutinated. Here, just accumulation, no mesh forms. It's negative. So agglutination we can do qualitative and quantitative. Qualitative means this positive and negative. Quantitative means we can say the theta 102, 104, 108, 116. So theta. So it's high theta compared to here. It is here up to 1 in 64 in patient 1. Here 1 in 18 patient 2. So positive control always VQ. Hemagglutination only difference is we use red cells to test. Hemagglutination inhibition is with the presence of antigen, hemagglutination will be inhibited. So that way. Positive test, virus, RBC, TET serum, no hemagglutination. So that particular serum contain hemagglutinin or TET serum. In negative test, viral, RBC, test serum, if there is that particular antibody is not there, so hemagglutination will happen. Complement fixation, another term, just to know. It nowadays it rarely for fluorescent antibody test we perform, direct fluorescent antibody test and indirect. Only thing is they elicit fluorescence light. So with the presence of fluorescence, immunofluorescence microscope, we can detect. That's it. So direct and indirect. So group A is streptococcus there, fluorescent dye-labeled antibody to group A. They form, then we can see through immunofluorescence mic. But indirect test, we use another antibody. Initially, this is Trichoderma pallidum, specific antibodies in serum of patient. Then, and once antibody antigen reactions there, these specific radiolabel antibodies go and act there. So we can see this. Other important thing is, even though serology, there are multiple limitations because we detect, and if we detect antigen, it's more towards diagnostic and helpful to management. But antibodies, and development of antibodies takes time. So in the initial stage of the acute condition, we can't detect antibodies. It takes some time. Other things we have to compare acute serum now for this case patient person, and we have to wait another 10 or 14 days to take conversion serum to detect rising titer of antibodies. Initial stage 108, maybe one doubling of 4 times rising of the titer indicates that patient has had this particular condition. That's how. So we have to wait. But in that particular period, patient survived okay. Otherwise, we can't go for the targeted therapy. So it's not helpful for management as well as diagnosis. Only for the chronic diseases like HIV, rabies, it may be helpful. So that's what I said earlier. And also there are molecular mimicry and cross-reactivity. HSV with varicella zoster, Japanese B with dengue. So specificity is low. It leads to false positive results. Problem with serology, other health condition interfere. So if patient is immunocompromised, they can't form antibodies. Infectious mononucleases, those with connective tissue disorders such as SLMME, non-specifically giving false positive results. So there are a lot of cross-reactivity. About ELISAs. So it is immunological methods, new methods, immunoassays, enzyme link, immunosorbent assays. Use antibodies linked to an enzyme link. And with these reactions, we can detect with the mass spectrometer. There are direct and indirect tests are there, human there. Different ways of performing ELISA, indirect and competitive and sandwich. But just to know, this is how it works. There are other chromatographical tests as well as in-house tests as well as radioimmunoassays. It's only for the instead of enzyme link of antibodies, here radiolabel antibodies is used. That's the difference. And also immunoglobulin or Western blot, technique separation of detection of proteins or antigens. It's helpful in diagnosis of HIV in serum, P24 antigen detection. Likewise, there are a lot. HIV difference confirm HIV 1 and 2. About the molecular assays, there are again different molecular assays. It goes with DNA or RNA detection. But still nowadays proteomics, protein detection is also there. So, but molecular assay, it's a genomic. Proteomics, small detox, like there are a lot of proteomic images. Genomics, that's DNA, gene tip, microarrays, likewise DNA and PCR. PCR, restricted fragment length, polymorphism, and genetic probes. Molecular diagnosis advantages reduce reliance of culture, faster, more sensitive, more definitive, more discriminative. Techniques adaptable to all pathogens. Disadvantage technically demanding, relatively expensive. Can be too sensitive, provides no information if results are negative. Other future. About the future, rapid diagnostic are very much important. Detection of rapidly, because in conventional culture, even up to world has developed, but it takes maybe 4 hours to detect those bacteria. Like we use Vitek2 platforms like this, but still it takes time. And also immunochromatography tests are there, like we are doing HCG testing. Here they are for the fat, or for the detection of malaria, filariasis, or influenza, there are fluorescent tests. Sorry, fluorescent immunochromatography tests are there. Another important aspect is laboratory automation. That means without use of human force or human labor. Now also is still under development. These are novel automated platforms, MOLT, metric associated, laser desorption and ionization, time of flight spectrometry. Nowadays it's used. And expert MTB RIF assay, detection of rifampicin resistant gene. It's also under there. Thank you.